Firstly, I'd like to acknowledge the traditional owners and custodians of the land on which I am today, the Wurundjeri Woiwurrung people of the Kulin Nation, and pay my respects to their elders past and present, as well as to elders from other communities who may be with us here today. My name is Julia Cirillo and I'm the Water Watch and Rapid Response to Litter After Rainfall Programs Coordinator, bit of a mouthful, at the Merri Creek Management Committee. Today I'll be moderating our Funginar, or Fungi webinar, the first webinar event co-organised jointly by Merri Creek Management Committee and Friends of Merri Creek. And I know we've got quite a few members of Friends of Merri Creek here today with us as well. Thank you. And thank you to Anne for organising this as well. Today, our presenter is Dr. Sapphire McMullan-Fisher, who is not only a very knowledgeable mycologist or fungi scientist, but who also lives close to the Merri Creek and walks there often. So she knows the fungi of the Merri well. And I know that I've seen Sapphire down the creek when I was doing some water bug sampling in spring last year, and we had a bit of a chat while she was going for a walk. As I mentioned, we'll be splitting this webinar into a presentation. So Dr. Sapphire will start now and then questions and answers. The Q&A will include Sapphire providing identification of fungi photos that people have put up on the iNaturalist fungi map. And of course, Sapphire will talk about the iNaturalist fungi map as well as answers to questions that arise during the presentation. So thank you so much, Sapphire. I will now let you share your presentation.
Excellent. Well, good morning, everybody, and thank you for joining on what is actually not a bad morning at all to be out and about, but here we are sharing on our screens. And thank you to both the Friends of the Merri Creek and the Management Committee for helping me do this. So I will now share my screen and tell you a little bit about fungi. So fungi are a kingdom all of their own. Unfortunately, some people still think biodiversity of plants and animals, but it is actually the diversity of all living life. Bios is Greek for living, so that is great. And here are some of the very pretty fungi that I've seen around the traps. So just a little bit of biology to help you understand why fungi are different in a kingdom of their own, and just to help you think that they are different. So some people think the mushrooms and puffballs and coral fungi are actually the whole mushroom. This is not true. Most fruit bodies, as we call them, or sporocarps, because they're where the spores, the seeds of the mushroom exist, actually are like seeing apples on an apple tree. If we only notice fruit trees when they had flowered and fruited, they wouldn't be there most of the time. And that's kind of where fungi, I guess, get unnoticed, is because they're actually in things, in wood, in logs, in leaves. Even in our creek, we have freshwater fungi that help recycle nutrients in our creek. They are in the things, so we often don't notice them. So not all fungi live as mycelium, which is the collective term for fungal threads, and that's what a lot of fungal bodies are. There are other different life forms as well, but today we're going to focus on the ones that do produce fruit bodies, and these are mycelium based. So they are these lovely threads, and we can't actually see them, but when fungi are starting to get ready to do something, so you can see this lovely mycelium here, and this fungus is, what it is doing is bringing all its nutrients together. And these are actually teeny tiny baby button mushrooms. And so fungi pop up overnight because they've done the work, they've drawn their nutrients together, and they've actually formed their little baby mushroom waiting for that rainfall to then expand. And that's pretty wonderful, and it is also why you tend to see fungi in the wet season. So we in Melbourne tend to have a wet autumn and often a wet winter, which we're being very lucky. We're having a great season right now, but it's not always the case. So up in the tropics, fungi pop up in their summer because that's when they get the rain with the monsoon. One of the awesome things about fungi threads, or this mycelial lifestyle, is that actually fungi can inhabit the same space. So they can inhabit the same log or the same patch of soil. And it's a bit like multicolored spaghetti. And they, like this bowl, they can all pass through each other and be lots of different species in the same space. And we only tend to, as I said, notice them when their fruit bodies pop up. The other interesting thing about fungi, because they are this mycelial thread-like lifestyle, is that they don't have a stomach. So how do they digest? Where do they digest? Well, they digest where they live, which is in their environment. And so it's I tend to use the colloquial, they have enzymes, they create enzymes, which they slobber out into the environment. These enzymes, a bit like when we chew and digest food, do that sort of mouth stage where we've got lots of saliva, the slobber, that actually starts the breakdown of those larger molecules, and then the fungus is able to absorb that, and that is how a fungus eats. So just to give you a little example, this is a slide from soil, deep in the soil, and these are the fungal threads here, and you see this loop here, and this worm-like creature is actually a nematode. Some nematodes are good in soil and other nematodes are bad and eat plant roots. When they're bad, we tend to call them eelworm, and this nematode is now actually fungus lunch because it has been trapped, it's gone through one of these hoops and been caught, and now these fungal threads will start to grow and exude enzyme and digest this nematode. And it's a little bit, because they're digesting out in the environment, it's actually a bit loose, so bacteria and protozoa and other organisms sometimes get in on the act, and that's one reason why fungi are great recyclers and sort of drivers of nutrition in the soil cycle, because it's actually a sort of open source food. I just wanted to just mention a couple of things that fungi do in ecosystems before we really get into the nitty-gritty of identifying some fungi from the Merri Creek. And so fungi, some people are very concerned about their environment, and they understand that our older, larger trees are wonderful resources because they have hollows, and people are very concerned about birds and some of the animals like possums that use these hollows as habitat. But what some people don't realize is that you don't just get an old tree, happy birthday, you're 100 tree, now you've got a hollow. It's actually a process. And pathogenic, which are fungi that actually do some harm to their tree partners, and wood rot fungi actually are the process. So a branch or something will break in a tree, a pathogenic fungi will come in and attack some of that living tissue and start living on it. But good pathogens, there are pathogenic fungi that have literally been living on their trees for decades to hundreds of years. So it starts to eat. When it's eating, it's mycelium. And mycelium is actually highly nutritious compared to wood, which the fungus is actually eating. So all these wonderful invertebrates, some people think they eat wood, and they may eat wood, but they're actually digesting the fungal hyphae that were digesting the wood. And so this is a start of a really important food chain, and there's a great diversity of wood rot fungi that start to come in. And once you get these small microinvertebrates, you tend to get birds and other animals that will come in and see this is a food resource. Insectivorous birds eat them, they hollow out the hollow, making it bigger. So we just need to be a little bit careful with our management strategies for trees, that we councils and other groups and homeowners tend to prune their trees and protect their assets, and this is all good, but we shouldn't prune everything all of the time, because then we'll actually be stopping hollow forming, and that's a problem, because then concerned citizens like yourselves run around and put up wonderful boxes to try and help these animals that have lost their homes. But this is from a picture from Jumping Creek, and there was actually a sign giving me the date of when these boxes were put up. So these boxes were put up in 1999, and this photo was taken in 2014 when I moved back to Melbourne, and you can see that we're only getting about 25 years out of these boxes, so they're not actually a long-term solution. So yeah, we need our wood rot fungi in our environments. They can also eat logs, so leaving logs and dead branches and litter is all actually helping the fungi recycle nutrients, which then it's like giving compost to plants. We don't want to run around in the natural habitat and compost everything. We just want to leave the fungi there doing their jobs, but it means we need to be cautious when we tidy up too much.
Sapphire, sorry to interrupt. We just had a question on that, actually.
Yeah, great.
On Rose. So questions already, and keep the questions coming, everybody. With the mycelium networks in the soil, is it best not to dig vegetable gardens?
Yes, as a rule of thumb, yes. The longer I learn about fungi, the less digging I do. And some of the digging suggestions come from northern hemisphere, quite young soils, where digging actually does release nutrients. But here in Australia, even on the basalt plain, this is probably some of the richest soils in Australia, but digging basically damages mycelium. And so if you have a good hyphal network, ripping and tearing out the plants is actually meaning that the next generation have to repartner rather than one mycelium being able to partner with lots of plants over generations. It's I would certainly think it's much worse in turning over soil in natural areas. Gardens are highly managed areas and veggie patches in particular. So I can understand that lots of people do it and it's part of their process. And, it's not the end of the world, but certainly around fruit trees and the like, tilling can cause damage. And at a landscape scale, ploughing and tilling can be one of the reasons why we actually have depauperate, not enough fungi in some of our farmlands anymore.
Wow, fascinating. Oh, thank you. That's it for so far. Thanks.
Great. Okay, so moving on from wood rot and recycling fungi and pathogenic fungi, and to a really important group of fungi. So these are called Mycorrhiza, meaning fungus root. And this is where the relationship of these fungi and these plants happen. And so this is a teeny tiny pine seedling, and it has put down quite a large root network, which is kind of the spaghetti here. And then this white fuzz is this pine seedling's fungal partner. And the relationship allows nutrient access from the soil and water, and the plant shares sugars in return. And I've just got a little sort of graphic video here. Basically, plants photosynthesize. They're very good at catching carbon dioxide from the atmosphere and turning into sugar. And fungi are very good in soil. They've got good enzymes. They can get nutrients like phosphorus and other micronutrients that plants need. So they basically share. And 90% of terrestrial plants are mycorrhizal. So actually most plant groups have fairly primitive roots because they haven't needed to develop them because they evolved together. So plants and fungi came out of the freshwater creeks and started expanding onto land at the same time. And this relationship's been going since then. It's gotten even more interesting. Some people call the mycorrhizal network the wood wide web, and this is because we people have observed evidence that they knew plants somehow communicated together, and there is some evidence now that plants do listen and respond to sound, but most plants actually communicate via their fungal network. So one plant can be distressed because it's being eaten by some insects, and it will pass on that information through the fungal network. So it's not Wi-Fi. When I first did biology in the 90s, I was told that the plants communicated through W waves, but actually it's more like a can and a piece of string. Basically, this plant shouts and the fungi pass on that message through electrochemical signaling. And even more importantly for ecosystem health, these fungi can share sugars. So a healthy, larger tree can share its sugars down with the undergrowth, and potentially some of this at different seasons, plants in the undergrowth may be actually sharing sugars back to their trees. So when we mess around in ecosystems and start selectively removing plants, we don't necessarily know what we're doing below ground and how we may actually affect that system. So there's heaps to learn with understanding fungi. Now, these mycorrhizal fungi, in Australia, one of the really important groups of mycorrhizal fungi are called truffle-like fungi. And these are fungi that Australia started drying out about 35 million years ago. And if you're a mushroom, you've got to pop up into the air currents to spread your spores. But Australia began drying. There were winds, and there was actually selection pressure to not pop up on anymore and dry out Instead, these fungi developed a relationship with many small ground-dwelling marsupials, our bandicoots, our bilbies, our bettongs, and even some of our wallabies and paddymmelons also eat some fungi. And so these are the things that spread the fungi spores in the poop, basically. We call them the matchmakers between plants and fungi, because fungi mycelium doesn't grow very far. Trees are static, they can't move around unless they're in the seed dispersal stage. So these relationships, a healthy tree will have dozens of mycorrhizal relationships during its lifetime, and a large tree may have up to 100 relationships. And that is the strength and resilience. Lots of different partners. Some partners are better at getting water, some partners are better getting phosphorus. So healthy systems have a great diversity of fungi, and these truffle-like fungi are some of the best partners for our trees. So it's actually concern for the fungi that we've lost our little matchmakers running around. Melbourne is lucky. We do still have some bandicoots, but unfortunately not on the Merri Creek.
Sorry, Sapphire, just sorry to interrupt. There's just got another question.
Yep.
Great questions from Rose. So thank you, Rose, for holding up the chat so far. It's great. So just for a lot of the research that you've been talking about with the mycorrhizal fungi, is there a lot of this related to the research Peter Wolleben did, The Hidden Life of Trees? That was the question.
So yes, that book does discuss this relationship, and that is Northern Hemisphere evidence. And unfortunately in Australia, one of our big problems is that we are still relying on information from Northern Hemisphere fungi and plant community relationships to understand our local fungi. We have done quite a lot of evidence on truffles, and the particular evidence that in Australia the truffle-like fungi are better partners with the trees comes from up in the wet tropics. And so again, we're supposing this pattern occurs all the way around Australia, because all the way around Australia there is a predominance of truffle-like fungi, and we do have local evidence for that. But it would be good to know how the relationships work down here in our woodlands and our grasslands, and actually know for here, not just borrowing evidence from elsewhere, which is a good start. It's sort of scientific anecdotal evidence, but until you know what's going on locally, it's still supposition.
So we definitely gap in the research for some budding fungi scientists if they want to go down that field. Very good.
Yeah, assuming we can pay these budding scientists and so they can live.
Yes, very true. Thank you.
Funding is the big gap. There's lots of research gaps, and usually funding is the whole. So with conservation and global warming, it surprises me that we need to say that actually planting trees and vegetation is a great way to suck up carbon, but we do, and it is. But what we now know is that healthy mycorrhizal networks are actually carbon sinks. So this is another reason not to go around and damage any intact ecosystems we have, because they are net stores of carbon. So if you have trees and they have however X amount of carbon, the healthy soil network under them actually stores 70% more than those trees. So the bang for the buck is trees and their ecosystems and the fungi together is the biggest store and most stable store of carbon. So that's where we need to go. We need to start reintroducing healthy ecosystems. And in grasslands, let's not put up the trees because they're grasslands or grassy woodlands. Let's, fix things appropriately. I get very concerned sometimes on the basalt plain that people think trees are the solution to everything, and I love my grasslands. So we do have problem fungi. I did mention that there were pathogenic fungi that were part of the hollow process, but we do have problem fungi, and here are 3 of our worst fungi. Myrtle rust is an introduced disease from South America. We're very lucky at the moment. We only have 1 strain of myrtle rust. I say lucky in the sense of we could have several strains. It's a real problem even with the strain we have. This is affecting rainforest plants and Melaleuca and Callistemon communities in particular. Luckily, all Victorian outbreaks have been eradicated. But you, as people, if we ever do get out of Victoria again, as travellers, you need to not wear hats up in Queensland and then bring your hat back and wear it down the creek, because this is how rust spores spread in air currents. So cleaning your shoes are no good for rusts. And basically, we should just, as human beings, stop moving stuff around. Crazy ants, fungi, all sorts of things, viruses that affect humans. One of the reasons that these things move around is because humans move around and they're not necessarily clean and healthy. So we can't do much about our viral health, but we can do something about our shoes, hats, and equipment. The other two fungi here, this is the orange ping pong bats, Favolacea. It's a weedy fungus that pushes out local fungi, and unfortunately in the 90s when it first arrived, we didn't do anything to prevent it, and now it is in lots of places. But if you ever see this, and lots of people are starting to photograph it because it is really pretty, when you come home, please clean everything. Clean and dry equipment and clothing and hats means that, yeah, you're not spreading things you don't know about as well. And the classic fairy book mushroom, the fly agaric, is also a weedy fungus and has in some places jumped to eucalypts and Nothofagus, our Myrtle Beach. So again, some kids love kicking these around. Clean their shoes. We just don't want to be spreading weeds.
We've got a couple of questions, Sapphire, too, if I can ask you as we go.
Yeah.
So Elizabeth asked, do we know what happens with fungi after a bushfire?
The thing with fungi is they're a whole kingdom, so different groups respond differently to bushfire. And I do have a whole talk on fire and fungi, and some fungi have evolved to reproduce after fire, and these are particularly important because some of these are actually mycorrhizal, so that fungi their mycelium, actually some of these ones that are going to respond after fire, their mycelium grows, and that it actually helps prevent erosion, which is really important to keep the ash bed in place and not let it run downstream and cause problems. And then they fruit. It's called a discomycete flush. So there's these wonderful orange and brown and black discs that are there holding the soil, and they sporulate in that after the first rains, because this is when seeds will be germinating, and this is where the mycorrhizal relationship, the partner between fungi and plant, can join up, is in that ash bed. There are other fungi. Some fungi are very sensitive to fire, and rainforest species in particular. So our Nothofagus and some of our riparian zones actually are the fungi are adversely affected by fire. And there are actually some fungi that both respond to fire but also need large woody debris. And so they are actually adapted to both fire and long unburned periods, indicating that Australia has had fire for a long time and interesting fire cycles. So there you go, a potted summary of my talk.
Yeah, I think definitely one for another time. More information is fascinating. Wow, you're doing really well. Another question from Anne: if you cut up the mycorrhiza into 10 pieces, do you get 10 fungi or just 10 dead fungi?
10 dead fungi. It really depends. Mycorrhiza are incredibly hard to transport. So the most evidence is that if you damage the mycelium, you kill the fungus. There some wood rot fungi and other ones that are, basically live in something the size of a wood chip. Some of those are transportable, but most of those are wood rot fungi and not mycorrhizae, and not mycorrhizae. So yeah, moving mycelium is fraught with peril, and we recommend different ways of moving fungi around.
Well, there's a couple of questions on that. So someone's just asked for some of those introduced ones. Should they be removed if you see them?
So with the orange ping pong bats, this Favolacea, some people, I have met people who have basically...
And that's the middle picture, isn't it?
Yeah, this one here.
Yep.
So this is really small. This is like the size of your little fingernail. So it looks big and beautiful here, but you're looking for teeny tiny orange dots.
Yeah, I've seen them. They're beautiful. They do look beautiful.
They do look beautiful. Unfortunately, I hope you cleaned everything, or you'll clean them now. So yeah, it's down on the Yarra Flats. I haven't actually seen it on the Merri Creek yet.
I've seen it around Elphington wetlands.
Okay, yeah. So, and this is the thing, I, you know, even though I try and be clean when I go into the sort of nice bush, I was walking down Yarra Flats and I saw it and I thought, oh wow, this is the first time I've cleaned my town shoes in ages. So, yeah, if you are a landholder and you get the infestation early, removal and burning or soaking into anaerobic mush, there are some options, is possible. But I think it's one of those areas of we really need education, so people, when they do go to the more pristine parts of bush, probably, we've lost the plot in Melbourne and probably the Otways, but we might have some hope in some of the other parts of Victoria to keep it out. Erranundra Plateau and a few precious gems perhaps, but probably we really should have done something in the 90s, not kind of 20 years later when it's got infestations in lots of places. So yeah, it's like any weeding. If you let it seed, you've got problems, but the removal of fruit bodies can be problematic. And the question is always, what do you do with them? Certainly do not put it in your compost. That's a great way to spread fungi.
Well, and actually, just more on the spread and reducing the spread. Nicole was just asking, what's the best way to clean shoes, hats, clothes, cars, to reduce the spread?
So you can use PhytoClean or 70% methylated spirits diluted, and that can work if you've got muddy shoes and the like. Just washing and cleaning clothes and hanging them in sunlight is definitely helpful, but you kind of hope that 99% of stuff is cleaned. Clean and dry is the best thing we can do. I think people get quite lazy when they spray meths around and PhytoClean and some of those things. It's like they don't have to actually clean their shoes. There's a magic chemical in there, and it certainly helps. But not moving that mud and soil, and not moving cleaning, washable hats. I now have, I've got some lovely felt hats that are wool, and I know I can't really wash them, so I don't wear them. I've got my Victorian hats and they don't go anywhere else, and they also don't go into the really clean bush if I've been wearing them around in town.
That's a good point. Is it be selective what you're wearing and perhaps look but don't touch?
Yeah.
We've got a couple more questions here, if that's okay.
Yeah.
So Rebecca's asked, are there good fungi that the bad fungi could be confused with?
Yes, we have a native Favolacea, which is white and quite rare. The Amanita muscaria, I'm hoping, is pretty recognisable. Myrtle rust, certainly there are other rusts, and rusts are quite hard to identify. But I think one of the wonderful things we now have are cameras and our phones. So instead of, in the old days, people used to pick mushrooms and bring them to me, and now they just bring me their phone, which is hygienically way better. And if you do have any concerns, unfortunately, the federal government has just cut biosecurity funding, but we still do have Victorian biosecurity services. So yeah, particularly myrtle rust, if you have photos of that, tell someone.
Yeah, that probably relates to this last question here a little bit too. So Emon's asked, is it true that one should cut mushrooms instead of picking them out with the substrate? There is much debate about the correct method in the psilocybin, sorry for the pronunciation, community, and he's asking for a friend.
Yes, so it really depends on what you're trying to do with fungi. If you are collecting them, if you're collecting them on public land, you actually need a permit, which I don't think it's ever been like foraging happens all of the time, but you actually need landholders' permission, and public land, you're meant to have a collecting permit as well. And yes, people who do regularly eat mushrooms, whether they're growing them in a kit, which is my recommendation, or collecting from the wild, cutting and leaving behind the base is very useful because you're not disturbing the mycelium. But if you don't know what you're doing, it's very risky because some people have cut their thing they think is edible and left behind the evidence, which is a vulva. So we do have the death cap, unfortunately, in Melbourne if people are foraging. So you always have to know what you're collecting and eating and have checked every single fruit body, because people regularly collect things they all think are the same thing, and actually because fungal myceliums can grow together, you can literally have two mushrooms touching each other, growing next to each other, and be totally different species. So some caution there.
Oh, thank you, Sapphire. It's great. And actually, that reminds me of a good story in my family when when I was young, my grandmother would go out foraging for mushrooms, and yes, unfortunately, one day picked the wrong ones, and my poor grandfather was throwing up everywhere. So even though she'd been doing it for many years, people do make mistakes. So it's a
Yeah.
really good point. Yeah.
Yeah,
Thank you.
and it can be very problematic, which is why there are some really good mushroom kits now. You may as well grow what you know, and also kind of know, some of that roadside collecting. I just go, really, diesel?
I know.
Delicious.
That's the other thing, isn't it? What else are you eating? Very good point.
Yeah.
That's great. Thanks. Oh, and we've already had people saying thank you for your excellent answers, comprehensive answers. So that's it for the moment. Thank you.
Good. Okay. So this is the Merri Creek guide, or just a couple of screenshots. So this is a PDF that's downloadable on the website, or you can just look at it on the website. And what I'm hoping is that this will just get you started spotting your local fungi and also knowing what to do with them, which is what I'll move into, which is how to share a record on this application. So iNaturalist is a natural history application. It has both, it's a web platform as well as a smart device platform, which I really like to be able to go in using my computer for some things, particularly searching and identification. But the convenience of taking some photos in the field and then going back home when I've got Wi-Fi and popping up my records is great. But I know a lot of people, they need to see something they've seen before. So hopefully this guide, there'll be some pictures that pop. So we've got a couple of pages of mushroom-shaped ones. And then we also have some other shaped fungi. Not all fungi are mushroom shaped. And then we also do have 4 pictures of some likely rare and threatened fungi, or lost fungi, that we would be very excited if we get records of, because we don't know as much about the rare and threatened fungi because they don't pop up as much because they're rare. So yeah, if you do want to just have a bit of a look at this guide, you can print it out. It folds up into a nice little A5 booklet if you get your PDF and just hit booklet. And so this is a screenshot from just this morning of looking for fungi, and this is a patch of the Merri Creek. This is the Market Garden, the Ceres Garden, and this is Harding Street, so Coburg's here. And so you can see these are basically little records, and you can see in this part of the screen, it's actually showing the different fungi that are popping up. And I will go live at the end of this, and we can actually look at people's records. So just to tell you about the Fungi Map project, iNaturalist is a whole community of, you can put your bird observations, your frogs observations, you can do both calls and photos as evidence of what you observed, though observed kind of means heard or observed. And so there are different projects. And so the Fungi Map project has a network of well-known local people who help confirm what you saw. So you can actually put up stuff that you don't know. And the other wonderful thing about this application is it uses computer-assisted identification. So the first photo that you put up should always be your most recognizable photo, because then the identification algorithm makes an educated guess of what you saw. And when we first started using this app about 5 or 6 years ago, it kept suggesting Northern Hemisphere fungi because that's what it had been trained in. But once we clocked about 5,000 Australian fungi records, it began guessing correctly for Australia. And now there's over 30,000 records for Australia. So it's getting very good at guessing our local fungi. So the combination of computer-aided guessing and then humans who can check it. So I'll just take you through the steps of making an identification or putting up a record. This is the endpoint. So basically you end up with a map. This is me a couple of years ago, and this is where I was out near Tulangi. This is a brown mushroom, which usually I don't recommend people like try and identify brown mushrooms. They're a bit like brown birds. They all look the same. But this, with these lovely fluffy scales on the young ones, is actually a recognisable brown mushroom. I've actually popped up a couple of pictures, and I'm just going to say note research grade, and I'll get back to that in a minute or two. So that's where we're aiming for. So I took some photos, and do take photos with your phone, or if you are taking small fungi, if you've got a macro application that will help the phone take better macro photos, that's good too. But don't use you can actually take photos through the app, and the quality of the photos aren't as good. So take your photos, and as I say, have your geocoding on, let it know your location services for this application. And then you can go home and have a cup of tea and upload your observations. And you can see that I'm a relatively balanced human being. I've got, a couple of nature shots, a few fungi, and even a vertebrate. So, very well rounded. I then go down and pick observe, choose the photos that I want. And I took a couple, oops, and I've done it again. This mouse is driving me nuts today. My apologies. So here's the two photos that I put up. And although this is cropped out a little bit, this photo was taken because there's an older, mature mushroom. So my first photo was the most recognisable. My next photo showed something else. And I always also try and encourage people to take a habitat shot, because a lot of fungi are habitat. Identification kind of bits of our brain are based on habitats. Like if I'm in a rainforest, I'm more likely to see X. Then this is where the algorithm kicks in, and if you hit here, it will make the suggestions of what it thinks you saw. And you then, if you're going to add it to the project, you need to join the project before you try and add it to the project. So I did that via my computer because I'm not very good on my phone. And so the Fungi Map project asks you 4 questions, but you only have to answer 2, which is what the fungus habitat was and what it was on. So on the ground, on a bit of wood. I'm quite verbose because I'm trying to capture as much data as possible. And these last questions you don't need to answer anymore. They're optional.
Oh, quick question, Sapphire, just on that. Ellen just asked, do you want gill pics as well?
Yes, and I will talk to that in another screen or two. But yes,
wonderful. All right, we've preempted you. Sorry, I preempted you.
No, all good. Gill pics are very, very, very good. So then, to get to this add project bit, you actually have to scroll down to the bottom of the screen, and then you hit upload, and then you can see here my record has popped up. Whoops, and now we're back at the beginning, but this is actually when I've just done it, and you see here it says needs ID. So there is a sort of verification stage in the iNaturalist that basically says 2 or more people, basically 2/3 need to say it's the same thing before it can become research grade. And we want it to become research grade because the research grade data goes to the Atlas of Living Australia, which is our national amalgamation tool for biodiversity data. Of course, we only want research grade for things that are correct, which is why we have these human checkers. And so, yeah, we can share the fungi and we can see them as they pop up. And I will just tell you a couple more things about the app before I move on more about data. So when you save your observation, you have 3 options. The default is open, so anyone can see when and where you were somewhere. So you can see where I was in Tulangi back in 2018. You can choose obscured, and this hides your observation. When you go in, it drops it in the right place, and any project administrator that you share it with also sees it correctly. But it only the fuzz is only just over 500 yards. So that means that if you're observing stuff in your garden, people don't know, there's a concentration of your observations in one place, is probably your house. But you can also choose private, which then only you or the project administrators see the correct location, and everybody else basically gets a 1 degree fuzz. So it really just drops somewhere in Melbourne, northern Melbourne, for me. So
just a quick question on that, on the app, Sapphire. Mary just asked, just to clarify, when you download the app, I assume you agree to join iNaturalist Australia.
Yes, there will be all of the agreement and
Yep.
stuff.
Sign your life away. But yep.
Yeah,
It's all good. It's all good though.
Yeah, we apparently share lots of data all of the time. I'm always endlessly horrified at yeah.
Yes, I think that's the thing with the advantages come the things
Yeah,
that we think
yeah.
about. I don't want to say disadvantages, but yes.
Yes,
Thank you.
it's a strange new world, but we're trying to survive. So here's the gill question. So in the bad old days, lots of people would dig up mushrooms and show the gills, and that is still relatively fine. Probably disturbing one mushroom for a confirmed identification is valuable, but you can see here the alternative is to use mirrors, and it does take a little bit of time and energy to get the trick of using a mirror and getting a reasonable reflection so that it's an identification aid. So you can see here's our little mushroom. It's a small mushroom, so the mirror is actually harder to use because there's less to reflect. And it was really low to the ground. I usually try and get a bit of stem in, but wasn't possible in this one. But this is actually a Leucaria that I just saw up in the local, up in Merri Creek. And I'm very excited about this because I've seen very few mycorrhizal fungi on the creek, and this is one patch, and there were actually 3 mycorrhizals that morning. So really impressive. 2 of them were mushroom shaped and 1 of them wasn't. So yeah, exciting to see. So that's the mirror trick. A little bit more on the Atlas of Living Australia. So this is our federally funded biodiversity amalgamation tool. And you can see that it's got a lot of records, and for birds predominate biodiversity data. I've seen a number of articles recently about how the bird observations have gone up during lockdown. And so you can see there is a vast difference between over 8 million records to under 100,000 records of fungi. And there's not even 1,000 bird species yet. And yet we've already got nearly 3,000 fungi for Victoria. And I would expect probably Victoria's got somewhere between 5 and 10,000. So we really have a long way to go to catch up. And these observations of birds are over probably 100 years. There are very few fungi that have been observed before really digital cameras. The number of records are very low. So we don't have good baseline data of what it was like before disturbance. So this is also just a quick jump in using the Atlas of Living Australia to give you some idea how it's useful. You can see this is basically just a 5K radius covering most of Coburg and Preston, and you can actually see there's a bit of a concentration along the creek because this is our largest patch of bush, and you can sort of see those bush patches because that's where most people are making observations. And you can see that animals win, but just there's nearly 1,000 plants, but fungi we still, and this was this morning, only have just over 100 records for this quite large area of a good number of suburbs. So if we want to know what our local fungi are, we actually need people recording them, even if they don't know what they are, because then we can learn. And the best way to manage something is to understand it and have good information on which to base your decisions. So I really encourage you to have a go. And just a 2-second what I do, I'm writing a fungi land management book at the moment. And this is part of my efforts to you guys to understand your fungi. And I'm now going to stop sharing for a second and change and go to the internet, and then we can actually look live at the fungi of the Merri Creek. This is the iNaturalist platform, and this is the computer-based platform. And you can see that here I have put fungi including lichens. And I've actually put a bit of a bounding box because otherwise we had an enormous number of records coming up, and I'm hoping this bounding box will stay. And so we can sort of scroll along and look at different records. We can also see the most recent records that have been put up. We're really excited. You can actually see a predominance here of this green staining coral, which we are very interested in getting more records of, because we do think this is one of our likely rare and threatened fungi. But I'm working with a community group called My Community, and we are going to do some DNA on this fungus because there's actually a small chance that it's actually a recent introduction, and then it could actually be a Northern Hemisphere weed. But there are records from really good natural patches of bush, which is why we think it's one of our locals that we want to know more about. So, is there somebody here that actually did already put up a photo so that I can jump in and look at their record in particular, or shall I just sort of move up and down the creek and discuss some of the identification?
So let's see. We have, oh yes, someone here has put something in because I had about 4 or 5 people that said they did. So Craig Burgess says his username is Craig Bees Trees.
Okay.
Craig Bees Trees.
So I then go to observers and scroll down, and it's probably one of these people that has
C for Craig, but that's not in alphabetical order, is it?
No, it's based on number of records. So yeah, the best thing to do is to actually send me the direct link. But I'll see if I can find Craig.
Yeah, so it looks like it was 3 separate words, but it doesn't look like it separates it there. Craig and then bees, as in the insect.
Yep.
I'm just and then trees.
Yeah.
Oh, but it goes on when. Is it time thing? Does it? Yeah.
Well, it's number of records, and then we've got that one's an interesting.
Oh, Craig's going go down. Oh, he goes there. It is. So Maggie said there it is. There's one record.
One record.
Oh, entry 83. Apparently it was. 83. Thank you, everybody.
There we go. There we go. Craig Bees Trees.
Great work, everyone. Love it.
Excellent. So yes, here's a little 101 on people seeing observation. So here's your observation. So this is what we really want. Oh, lovely. So this is a little Earth star. So we can see the location, and we could zoom in and go exactly beautiful. Down near Clifton Hill in Yarra Bend Park. Lovely. And now let's go into the record. Oh,
Craig, that's beautiful. Great photography. Wow.
Yeah, and a couple of different photos. And so these guys, as the name suggests, Earth star, and that's really what they look like, a little star sitting on the ground. And you can see here this is called a plicate mouth, and this is the spore mass. So this is these guys puff their spores out of this little opening, and so we know that this is one of the plicate mouth ones. The other characters I'm looking at here, you can see this cracking and quite a relatively thick tissue for this Earth star. And some of these Earth stars are rather cute. They're hygroscopic, which means they fold up when they're dry, and then they expand out like this one and really stand up when they're all wet. And so I am not very good with my Earth stars, so I absolutely know, yes, this is an Earth star at the genus level, so Geastrum, but I don't go down to species level, but I can confirm Craig and Roxanne's ID of, well, Craig just put it up as fungus, and then Roxanne 67 said Geastrum, and I'm agreeing with Roxanne, which is good. And perhaps one of our Earth star experts will be able to recognise it. The only other photo I would have got, Craig, if we go back up, is you see how we can almost see if there is a little neck between the bottom of the Earth star and the start of the ball part. If that's the case, I do know that that's an important feature, but we can't quite see it. I'm guessing, just based on the distance here to what looks like the base, that there probably is a neck, but if you could get a side-on shot, that would help too. And possibly a scale, because some of these Earth stars are incredibly small, And so the 5 cent piece, the echidnacide out, is a really nice kind of cute Aussie scale and quite good for some of these small ones. But people don't carry coins as much.
No,
So
especially can't use them much at the moment.
I have a few fungi friends who literally have 5 cent pieces strapped to their phone.
There we go. That's a good use for them. Craig's just said they're small, less than 1 centimeter diameter for the ball.
Yeah.
And he said he'll next time have a closer look. And
yeah.
also, yeah.
And then this is a great thing is learning the different groups. And so I'm a real Jill of all trades. I know my Australian fungi quite well, but I certainly know some species and some groups better than others. But ecologically, be really happy with genus, because genus helps tell function. And so often, so a lot of the geastroms are actually mycorrhizal. I think there's a few that might not be, but yeah, so it's good to, I used to think when I first started in ecology that it'd be really easy, like all the puffballs would be one job and all the mushrooms would be another job. And no, unfortunately, you need to at least know genus and have had some science done to know what job they're doing in the ecosystem.
So Eamon's been saying he's been seeing these everywhere in the last month or so.
Oh, cool.
Yeah.
So yeah.
Just sorry to interrupt, Sapphire. We are at 12 o'clock now, though, so I do know that some people may need to go.
May need to drop out. So yeah, I'm happy to spend another 5-10 minutes and just do a few more identifications so people can get the gist.
But as long as hopefully everyone's happy with that. If they have to go, they have to go. Yeah.
Yeah.
Okay, because we did have someone else here. A couple of people have put up their usernames if you wanted to check them. Yeah.
Yep, we'll go back out and go back out.
So let's see.
Go back
out. So we've got Daniel, his username is Marsden. He said his photos were nearer the Derribin Creek, Yarra and Fairfield. Is that okay?
If I scroll through and he spots his name, because probably going to be a lot better at picking his name than me, but I am looking. Or we can go
number 6. Mary said. I think Mary saw number 6.
Okay, all the way up
for Daniel.
There we go.
There we go. Thank you so much for that, Mary.
Good spotting. Oh, excellent. So yes, again, the map, we can see that where these are occurring. And if we just go through, so these guys are lovely, and this one's reached research grade. So these is a polypore, so it's not soft like a mushroom, and these hang around for quite some extended amount of time. They're really good wood rots. They're very strong, so always be cautious. If you ever see this on structural wood, you actually need to do something straight away. And here, lovely shot of its pored undersurface. And I'm going to try and just zoom in. Yeah, so you can actually, now that I've zoomed in, see these lovely little dots. And the spores actually drop out of these lovely holes. And this is in beautiful, lovely, fresh condition. So probably when these cinnabar or scarlet bracket fungi are fruiting, spreading their spores, that's this lovely bright stage. And luckily in Victoria, most of our cinnabar polypores are the same species, but the further north you go, the taxonomy is just being done at the moment. So, and this used to be known as Pycnoporus. There's a global shift towards Trametes, but it's a bit of a taxonomic discussion. So very recognizable. You can go around the world and recognize this kind of group. Let's go down a little bit. I'm trying to pick the more recognizable fungi so people
just while you're doing that, Sapphire, Gail's just asked, how necessary is a microscope for fungi ID? Do you have any suggestions for getting a second-hand one?
Well, certainly if you really want to get into your fungi ID, a microscope does become necessary. So probably we think about 30 to 50% of fungi may be recognizable from photographs, and that is only once a taxonomy has been done. So that's quite a lot. If we've probably got, 5 to 10,000 macrofungi in Victoria, and somewhere, nearly half of them might be recognisable from photo alone, that's not a bad number, of things to just take photos of and get to know. But certainly, yeah, a microscope. But where you get secondhand ones, universities sort of used to get rid of their microscopes, but the less hands-on we get with science, I think the less microscopes there are in universities, and they don't get rid of them anymore. So I haven't heard of a university getting rid of a big batch for absolutely ages, probably through 13 years. Probably
Yeah,
was
it's
the
true,
last one.
isn't it?
Was the
Rebecca's made a good suggestion too. And when I was thinking this too, you got your tiny fold-out microscopes, or more of the lenses that can
yeah,
throw in a keyring or a necklace. And
yeah,
she
yeah,
got
no,
that off eBay. Yeah,
a
that's
hand lens. Yeah, hand lens, and I've just got one of these clips to go on my phone to give me more macro shots. So just to go into this fungus, so that you saw how much I had to zoom in, and so this fungus's common name is the split gill, and this is what I want to show you. And I'm not quite sure how I've managed to get the photo moving up and down, but the gills here are actually bifurcate or split. So I'm going to zoom out again. So yeah, they're very small, and these are little, they're attached to the underside of wood. I'm going to see if I can go back out, escape. There we go. And there was a second shot. And you can see here that these ones actually have a little bit of a stem, but again, lovely, very small fungi. The split gill, recognisable, cosmopolitan, so it's found around the world, and we don't think this is a weed. We think this was always found around the world. And just a little bit of caution. This is one of the fungi that do have a risk with them. So don't sniff the gills of this one. If you're immunocompromised, unfortunately, there have been a very occasional infestations in people's nasal tubes of this one. A bit like, so treat it like you would dry potting mix. Don't inhale it. There are a few risks, but there, on the whole, getting out in nature is much better for you. And so we hope we just get the good health from that. I'm going to go down. Oh, lovely. Where did you see this one? Is this one local? A little bit further out than the Merri Creek, but still, let's go in. So Naghan's bonnet, so mycenas are these lovely wood and litter rot specialists. And, yes, we can see some of the little dots on this shot. So it's a really, they're little pixies parasols is sort of one of the common names for this sort of shape. And these guys have these teeny tiny white dots. And so when this fungus was first discovered, by Bruce Fuhr and Tom May. They were walking together, and there was a bit of a hollow log. And so Bruce looked into this hollow log and said, had a bit of a look. And Tom said, "What have you found, Bruce?" And he replied, "A Naghan." And there's a story of an Aboriginal sort of monster that had glowing silvery eyes and, would take people. And so the Cheryl Gravonovic did the taxonomy of this group. So it became known as Mycena Naghan for these little lovely little dots. And she did the taxonomy and kept the name. So yeah, they are glorious little.
And I'm sorry to interrupt, Sapphire, but we are nearly at 10 past
10 minutes. Yes,
12. So
okay.
I know, and I'm sure other people go, "Oh, Julia, be quiet," but I am just conscious of the time.
So hopefully people have now got a sense of the app, and hopefully will be brave enough to have a bit of a go, take some photos. Some really, really good observations. And so hopefully if we do this again, there'll be more than 100 fungi, within 5 kilometres of sort of our patch. As I think
you're certainly getting me enthusiastic to go out. And I did see that. I'm sorry, Eleanor, we won't have time, but Eleanor did say she had some things up on iNaturalist as well. But there's a couple more questions I just wanted to get to, because for some people are having to leave. So because John asked a question a while ago, and I want to make sure I don't miss him while I find it, while you unshare your screen if you like. What effects do introduced weeds have on our, I think he said our native plants. I'm assuming, or maybe he was asking about
Weedy fungi.
The weedy fungi, yeah.
Yeah, so this is where I really would like to see some research, because there are some products that are sold as fungal inoculants. And we do have quite a few weedy mycorrhizal fungi that are, well, introduced mycorrhizal fungi around. So one of the questions is, do weedy or exotic mycorrhizal fungi support the species that also came from the same region, the Northern Hemisphere? So a lot of our plant weeds are also from, the roadside herbs of Northern Hemisphere. Is there actually a beneficial interaction between those weedy fungi and weedy plants? We need the data. We don't know. But New Zealand banned the sale of these products that have these Northern Hemisphere named inoculums, because they realised, they did the research and could show that those inoculums were basically supporting weedy pines in New Zealand, and New Zealand spends an absolute fortune trying to keep the pines out of their lovely alpine zone. So they prevented those products coming in. Australia, you're still allowed to sell those products. Unfortunately, companies are a bit dodgy, so they're trying to continue to import them in New Zealand, but they're no longer calling them an inoculant. They're just calling them soil conditioners. So I think we really do need more information, good science about our local fungi and how our local fungi are helping our local plants, and if there is an interaction between weeds and weedy fungi.
Great. So we had another question here. Ellen was just asking, are there any visible signs of mushrooms that are dangerous to sniff?
Recommending you just never sniff fungal spores. If you are, some mushrooms do have a lovely odor, but if this is the mushroom cap, sniff this way, don't turn the gills over and sniff that way. Basically, that's your kind of safety protocol. If you're going to sniff them, sniff the cap, don't sniff the gills.
Someone's just asked, is there a way to get in contact with you, I think, to talk more about these types of things?
You can get to me, you can go into my Fun Ecology website or my Fungi for Land website, and I'm pretty sure I've got my email there somewhere.
Yes, did we put it down? Oh, no, we haven't. No. Yes, so it was on your presentation, wasn't it? Did you want to perhaps put that in the chat, or?
Yeah, let me find that. Was there a final question?
I'll have a look. I think there's lots of people saying it was absolutely wonderful. There was another question from Malcolm. Fungi for Land, is that a membership-based organisation?
No, at the moment we are a collective of mycologists and people who support us. And yeah, we're trying to write this first fungi land management book for Australia. It actually will be a global first, we think, because at the moment there actually aren't any fungal land management books anywhere in the world. And so my part-time job, currently for free, but we'll get there, is to finish the book. And we have 26 authors. We have 12 other Australasian mycologists who are going to help edit us. And yeah, we're trying to get the content ready so that we can actually get some plain English editors, because I did ask for nice, clear English and I got science speak from all of my scientists. Anyway, I will stop whinging. We're having a go at, getting the information out there, because I love talking to people and sharing these stories. But and I've just put in the chat my email for people.
Oh, wonderful. Thank you. I've also just put in the Merri Fungi Guide. So a few people are asking about that, and the iNaturalist Fungi Map, as well as some Merri Creek links as well.
Great. So yeah, together we can do better. We're doing good.
I can see lots of enthusiasm. It's fantastic. Is there a Merri Creek Fungi Facebook page? Are you aware of that? That'd be nice. Maybe someone needs to start it.
There is a Victorian fungi Facebook page, and there's some really lovely people. And so, yeah, if you want to go local, I think there are some smaller ones. There's a Mount Macedon one, but Yeah, I
There's a
don't.
gap. Why not?
There's a gap. I don't have the energy.
No, I'm sure someone here perhaps though might want to on this group. Please do and please invite us all, or I'm happy to send around the link if someone does make a Facebook page or some other platform. We've just got lots of people saying thank you, wonderful Sapphire, they love you. And someone said deaf fungi, not a moldy subject. Lovely dad joke, so thanks for that. Thanks, Anne, for putting up. Yeah, we actually have digital fungi jigsaws on the Merri Creek Management Committee website now, and she's put the link in the chat for those that want it. And I think I've grabbed everybody. I hope I have. I'm sorry if I missed your question because there was a whole lot of stuff going in at the last. Sapphire, you've just inspired everyone. And you've definitely inspired me as well. I'm very excited. I'm sure a lot of people are excited to get out there. And thank you to those that have been. Just some amazing photographs there as well. Was there anything else? I'm not sure if Anne wanted to mention. We will be having some other webinars coming up. I don't think we've actually, no, they're subject to confirmation. So they're not actually on yet. On our website, but if you are interested, please keep in touch with the Friends of Merri Creek Facebook page, and the Merri Creek Management Committee has a subscription as well. If you want to join the Merri News, you'll hear of any upcoming webinars. We are planning, I've got a frog webinar and there's a few others coming up in the next couple of months, so we're constantly preparing those, so please do subscribe if you want to be kept up to date with that. And if you've got any more comments or feedback, please put them in the chat, or you can email me if there's any if you need anything or any other questions afterwards. I'll just put my email in the chat for anything else in the future. Sapphire, was there anything else you wanted to add before we?
No, just thanks everybody, and yeah, have some fun spotting your local fungi.
And it was absolutely fantastic. Really, really enjoyed it. And everyone's saying just that. So Anne McGregor from Merri Creek, Friends of Merri Creek, did you want to say anything to finish off?
Only to say thank you very much to Sapphire. It was a great presentation. I'm sure we could have listened to her all day, as somebody said. And I would encourage people to subscribe to the Merri e-news and events, so you hear about future webinars and happenings along the creek.
Yes, thank you so much, Anne. Thank you, Sapphire, and thank you everyone for your enthusiasm. We can see that there's definitely a lot of interest here, so we'll keep the webinars coming. And Sapphire, absolutely fantastic. I also could have listened to you all day. I think we all could have. Thank you so much. Feedback, please do send it to me on the email or my chat there. And thank you everyone for coming. Have a lovely rest of your Sunday. Go and enjoy the sunlight, sunshine, and go and look for fungi.
Go out, do some spotting. Thanks, everybody.
Yes. Do some spotting. Thanks, everyone.